happy to be here and to have the opportunity to present to you a little bit about single-cell RNA-seq data analysis. So I'm going to talk about best practices a little bit today, and best practices are a joint effort for sure. So a few of the people I've learned from, I just want to highlight before we get started. In the data sciences platform specifically, I work with Murtash Babadi. He's the group leader of the group that I'm a part of. And he's taught me very many things about single-cell RNA-seq analysis. In addition, I've had the opportunity to work with the Precision Cardiology Lab and Patrick Eleanor's group. And from several very talented analysts in that group, including Mark, Carolina, Sikander, and Amelia, I've learned a great deal about single-cell RNA-seq data. In particular from Mark, with whom I've worked on several projects. If you have any questions about single-cell RNA-seq and you see Mark around the halls of the Broad, ask him a question. So as a general overview, what do I hope to cover in this talk? It might be a little bit packed, but hopefully we can get through an overview of single-cell RNA-seq analysis. I'll introduce the data, the experiment briefly, different possibilities for data analysis and challenges that are sort of unique to the single-cell aspect of this data set. And I hope to touch on a few key points in the pipeline, highlight a few things that I think are important, and have a discussion about best practices, bearing in mind that these best practices are still rapidly evolving in this field. I'm not going to be doing a step-by-step review of code examples. Unfortunately, we don't have time in this talk, but there are several great examples where you can go to see detailed code examples. Let me just highlight Tim Tickle's course from here at the Broad, the Hemberg Lab course, and a paper from the Theus Lab called Current Best Practices in Single-Cell RNA-seq, which is well worth a read if you have the time. Equally useful, I think, to take a look at our websites that have the documentation for some of these analysis tools themselves, in particular ScanPy if you're using Python, and Surat if you're using R. They have a lot of vignettes that are very helpful. Okay, so just to give context for this talk, in the past couple of weeks, we've heard some great primers about bulk RNA sequencing data. That type of data is what I like to visualize as a count vector. What you're doing is you're trying to count the number of times you see each gene in a given experiment. So you prepare a tissue, you try to sequence all the RNA in that tissue, and you count how many times you see each gene. You make this histogram, and you get a big vector of counts per gene. You can do a lot of interesting things with that, as has been covered, just to refresh our memory a little bit. One example is you can measure two different conditions, get a vector of gene expression for each of these two conditions separately, and then compare them. Using some kind of differential expression analysis, you can take a look at what's the difference between sample A and sample B. And you can see that there are some genes that are upregulated in condition A and some genes that are upregulated in condition B, for example. So conceptually, what's the difference as we move to single-cell RNA sequencing? Well, one of the ways to sort of visualize what we're doing is we want to separate out this vector from bulk RNA sequencing into its individual cell type components. So this bulk signal conceptually is a sum of these signals from the different cell types that are present in the experiment. So going from bulk to single cell is almost like going from black and white to a color photograph where you have all these different channels of information now. So why do we want to look at single cells? I probably don't have to convince anyone here that it's interesting, but just to review some of the main ideas, maybe we want to understand the biology of single cells. How do cells work? What makes one cell type different from another cell type? within a cell type, there can be a range of different cell states. How varied are they for a given cell type and in different conditions? Maybe we want to understand more about disease. Which cell types are relevant for disease, which are not, which play the major role? Is disease a change in cell state, cell type, or cell number? What exactly is the picture that's going on? These are some of the things you can answer with single cell RNA sequencing data. And thirdly, it's worth mentioning that differential expression depends greatly on cell type. If you do a differential expression test like I showed before on bulk data, your conclusions might end up being very different if you look at single cell resolution, where a trend that you thought was negatively correlated from bulk data could actually switch directions entirely if you focus on one cell type. And finally, another reason might be cells communicate with each other, and it's really interesting to look at cell-cell interactions, and we can do that by looking at single-cell RNA-seq data. There are several new challenges that come up when you're dealing with single-cell data, one of which is sparsity. So here you can see that as we move from bulk to this kind of summary of the single cell data, it doesn't look too bad. It looks like we still have a nice vector of expression for cell type A, and we have a pretty good idea of what's going on. But really, this is a summary that's painstakingly created from the raw data. The raw data looks a little bit more like what we have over here on the right. If we go all the way down to the level of individual cells, which is the data that we're getting in this type of experiment, the measurements are very sparse. We get maybe on the order of 10,000 counts or so spread out over maybe about 30,000 genes if we're looking at human data. So you can tell right away that a lot of these entries are going to be zero. And as a matter of fact, if you look at the data, the vast majority of entries are zero. And just looking at one cell is not nearly enough information to directly reconstruct what is the mean expression of the cell type, say. So somehow we have to infer this cell type information from all of these sparse measurements that have a lot of zeros. There are also new technical artifacts that come about in single cell data. We'll go over them very briefly later. So as an outline of the rest of this talk, I'll go over what is the experiment, just so we're all on the same page. I'll do a blazing fast overview of the analytical workflow first, so that we get an idea of the major points in the pipeline. And then I'll go back and revisit a few steps and talk about some more detailed considerations for analysis and some steps that I would recommend adding to the most basic workflow. And finally, a few concluding thoughts and recommendations about best practices so far. The experimental setup. I won't spend too much time here, but this is a basic diagram of, for example, a 10x genomics type experiment that's droplet based. The idea here is to capture individual cells within their own tiny droplet. So what's happening here is cells are entering this microfluidics device. They're being captured in these droplets of water and oil, and all the cells are separated from each other. And now the cells are these whitish dots here, and these colorful dots are barcoded beads. So each droplet gets one bead, and that bead contains DNA barcodes. These barcodes get attached to a cDNA transcript that is made from the mRNA that's present in the cell within that droplet. So as you can see, the idea is that within each droplet, all the mRNAs that were there get reverse transcribed into cDNA, and they get tagged with a barcode that is specific to that droplet. So all the transcripts in this droplet have the same cell or droplet barcode. That way, when we do bulk PCR and sequence, we can trace back and say which droplet each read came from based on the droplet barcode that it was tagged with. So that's the general idea for this kind of experiment. And this is just one method. There are others, but this is the one that I'll focus on today. The end product of this type of experiment is shown in detail up top. This is probably a little bit too much detail, so let's simplify it down a little bit to the key elements that I will mention further in this talk today. Those elements are the droplet barcode, a unique molecular identifier, the UMI. So this droplet barcode is the same for every molecule that comes from the same droplet, whereas this unique molecular identifier is different for every single molecule in the experiment. It just tags that this was one unique starting molecule. And those two things are attached to a piece of the actual mRNA transcript, which gets copied to cDNA. So this is a long piece of DNA that contains droplet barcode, UMI, and a little bit of the transcript gene of interest. So the data analysis pipeline, let me go over a typical workflow. And we'll just do this fast at first and revisit it. The most basic workflow is that you start with some reads in a FASTQ file, say, that come off of the sequencer, a big list of reads. You condense these into what's called a count matrix. We'll go over that. And then you take this count matrix and you begin the map making process, which involves dimensionality reduction, visualization, and clustering. There are several downstream types of analysis you can do with this. But the major pillars of this are per cell quantification and making a map.
Stephen, just to interrupt you with a question that came up in the Q&A. Thank you. The question, now there are two. The first question is, does the UMI come in the original droplet and what does it look like? And the second question, which I'll ask now because it's closely related, is how do you get the UMI to tag each individual molecule?
Great questions. Okay, so I don't have a slide that depicts this. I should have included a backup one, but so the idea is that on these barcoded gel beads that are painstakingly produced by this company 10X or others, this bead is covered with tiny little pieces of DNA, and those pieces of DNA have the same droplet barcode. So millions of pieces of DNA here. All of those pieces have the same droplet barcode, and they have a random UMI sequence. So what it actually looks like is 16 bases define the droplet barcode, and the following 10 or 12 bases define the UMI. And that UMI is totally random sequence. Exactly how that's prepared, I don't know. That is part of 10X's process to produce these gel beads. Probably one of the most important parts of the experimental procedure. And those two elements, the droplet barcode and the UMI, are followed by a poly-T stretch. That stretch of poly-T is then used as sort of a bait to allow polyadenylated mRNAs to hybridize. So you lyse the cell open within the droplet, all of the mRNAs spill out into the droplet, and the polyadenylated tails of the mRNAs hybridize to the poly-T capture piece of those barcoded DNAs. And then in the process of making a cDNA, you make a full-length transcript that has barcode, UMI, and transcript. I hope that answers the question.
Thank you. It seems to. Thanks.
Okay. So with this most basic kind of a workflow, the idea is to start with reads off the sequencer, you condense these into a count matrix where you're counting the prevalence of each gene in each droplet. So you have a big matrix here. And then during the map making procedure, again, we'll perform dimensionality reduction, visualization, and clustering, and we'll end up with some kind of a map that looks like this. And you can summarize the experiment with these neat two-dimensional representations of this complicated data set. What you can see happening here is every dot represents an individual cell from the experiment. And you can see that they sort of form these little groupings or clusters. And the idea in this picture is that similar cells are close together on this visual representation. And so what's happening here is the different colored blobs are actually different cell types in the experiment. So let's go through a pictorial overview. How do we obtain the count matrix? Okay, so briefly refreshing this library prep. What we have is these black outlines are droplets. We have single cells, each in their own droplet. What happens is we lyse these cells within their droplets. We reverse transcribe polyadenylated mRNAs, which have been attached to droplet barcode UMI poly T tail. And we make this construct of barcode UMI transcript. As you can see, the droplet barcode is the same for all the molecules within a given droplet. And the unique molecular identifier is different for each unique molecule that was present initially.
Another question or a set of questions has come up, and this is, I think, immediately after the slide you have here. So what are the lengths of the reads that come out? And at what steps do you lose the most molecules per cell?
Interesting. So the lengths of the reads, there are actually two reads in this process. Read 1 covers the barcode and the UMI. And read 2 covers part of the transcript. And that's typically like a 98 base read as far as the actual transcript is concerned. And sorry, what was the second part of the question?
Sorry, the second question, it was at which step or steps do you lose most molecules per cell since the data is so sparse?
Interesting, yeah, very good question. It seems to be the case that the step where you lose the most molecules is this cDNA step. So somewhere between trying to capture these mRNAs by hybridizing poly A tails to poly T capture oligos, somewhere between that process and actually creating the full-length cDNA, we lose some molecules. So of the maybe ballpark 100,000 mRNAs that might be in a cell, we see maybe about 10,000. So we're only seeing about 10% order of magnitude of what's actually present in the cell. Probably one of the main reasons is that when we make that cDNA, it's just as a technical detail of this library preparation step, we have to create a full-length cDNA that gets all the way to the end of the transcript in order to add on a piece that's needed for PCR. So unless we get to that full-length transcript, we don't actually see the product in our final library. And depending on how long the transcript is, some transcripts are very long. Depending on how long they are, the likelihood that we enzymatically get all the way to the end and create that full molecule, the likelihood goes down as the length of that molecule gets longer.
And there's one more question. Thank you. It's also on this general theme. So the question is, UMIs will by definition linearly increase with sequencing depth. So how much depth does it typically take to sample all the droplet barcodes, that is cells, within a typical 10K cell sample?
That is a great question, very important, and unfortunately I'm not prepared to answer it. I may have to tag someone in afterwards in the discussion section to help out with that question.
That sounds great, thank you.
Yeah. Okay, after we have these original cDNA molecules in each droplet, then we can break apart the emulsion, mix everything together, and PCR amplify all these molecules together in bulk. We grab some of them for sequencing, And then the idea is to take these sequenced reads. Now these molecules represent reads that we've sequenced. And the idea is to map them to a reference transcriptome and count them. So here on the bottom is our reference transcriptome, where we have all the bases in the genome here, and we have definitions about which parts of the genome constitute different genes. Gene A here, gene B here, for example. And what we can see is that when we map these reads to the transcriptome, some of the reads align to gene A, some of the reads align to gene B, and each read, again, is tagged with the barcode information about which droplet it came from. So we can demultiplex these reads into groups now, where we can say, droplet number 1, gene A, we had these molecules. Droplet 1, gene B, we had these. Droplet 2, gene A. Droplet 3, gene A. And on and on. And what we can do is take a look at the reads that we see. And for example, here we have 10 total reads, but only 4 groupings of these unique molecular identifiers. So the fact that there's just 1 of this molecule and 4 of this molecule in our final sequenced library is just due to PCR amplification biases, or which molecules we happen to sequence. It doesn't reflect that there were more of this molecule originally, because all of these 4 molecules are copies of the original molecule that had this unique molecular identifier. So, and to summarize this data, we count the number of unique UMIs. We take those numbers and enter them into this big count matrix. So the main object is to come up with this quantification, which is the unit of that is the count matrix for single cell data. And so we might have a big matrix that's droplets by genes. Maybe there's on the order of 100,000 or more droplets, of which maybe 10,000 or so contain real cells. And depending on what organism we're working with, if we're working with human, maybe we have about 30,000 genes annotated. You can see that there are quite a few zeros in this matrix. That's indeed what it really looks like. There are many, many 0 entries. Okay, in pictures, what happens with dimensionality reduction? And still, I'm going a bit quick, but we'll revisit some of these things in more detail. Dimensionality reduction, the idea is something like this. We have a big count matrix, which has a lot of cells and a lot of genes, but as you can see, many of the entries are 0. And it sort of looks like if these are the same cell type, there's some information shared across these things. In fact, if they're the same cell type, we expect them to be quite similar in some way. So a few observations. Most entries are 0. There are many genes that don't really vary much. They're kind of similarly expressed in many cell types. So that's another observation. Finally, we expect there to only be a few cell types in a given experiment. We don't expect there to be 100,000 cell types with 100,000 different droplets. We expect that maybe there are on the order of tens of cell types or less. So the intuition is that the hypothesis here, maybe we can represent cells with fewer numbers than the full 30,000 numbers that we need here for the full data set.
There's just a point of clarification also that I think is related to this. So one audience member asks, why do you use the UMI to count instead of the barcode?
Yes, so good question. Instead of, I guess I'm wondering which barcode. It can't be just the droplet barcode, because that will be the same for every molecule that comes from the same droplet. So you definitely need to separate these out, because there could be different original transcripts that came from the same droplet. But the unique molecular identifier tags what was initially one molecule within that cell. So the idea is to count how many unique ones of these we have as a proxy for how many original molecules there were. rather than counting the full 10 here in this case, which is more a reflection of maybe PCR amplification bias than it is of the biology.
Great, thank you very much.
Yeah. So we hypothesized that maybe we can represent cells with fewer numbers than the full 30,000. And the procedure that people typically use here is to somehow normalize and log transform the data, select highly variable genes. Again, this has to do with the fact that lots of genes don't really vary too much. We want to see which things are the most highly variable. And along with that idea of high variability, we actually want to highlight sort of groups of genes that vary together and account for most of the variation in the data set. You can do that using principal component analysis. This gets you down to, say, maybe, depending on your decisions here, maybe 20 to 50 dimensions. So now we're representing cells, instead of with the full 30,000 numbers, maybe with 50 numbers. Using that, we can define a notion of distance between cells. With this vector of 50 numbers, we can calculate a distance between this cell and this cell by looking at some kind of distance measure between those two vectors. And we can use that to calculate a graph of nearest neighbors. So cells that are similar become close neighbors on this kind of a graph. We can use that graph, we can partition that graph in order to do clustering. And we end up with the different colors that we see here. Further, we can reduce this 50-dimensional principal component space to 2 dimensions using an algorithm like UMAP in order to really get down to a 2-dimensional image that we can visualize here. And it would look something like this.
Stephen, there's another question from the audience member about the assembly of the count matrix. And the question is, are there many detailed post-processing steps to clean up the data before getting the count matrix, or does most of this happen afterwards?
Great question. And it leads into 2 of the things that I'm going to touch on in more detail. So I will cover that for sure.
Great, thank you.
Yes. Okay, so now we're done, right? So that jumped the gun a little bit, but just kidding, we're not done at all. There's quite a few things that we should consider in a lot more detail, as the question implied. So let's take a more careful look. What are some of the best practices? How can we do this carefully? And let's just get into a little bit more detail. Again, this is the most basic workflow that I showed before. I'm going to add 2 or more little modules here, and we'll see what I'm talking about. Let me highlight one at a time. First, we're going to talk about processing that happens before condensing things into the count matrix. And then I'll talk about cleaning up the count matrix once you have it. So first, I'll talk about trimming the reads themselves. Here's a general observation, something I came across when doing some data analysis as part of the precision cardiology lab effort. This is a screenshot from IGV of a pileup of reads that happen to align to this gene called LINGO1. I noticed that they were changing in number quite a bit in different conditions, and it seemed a little bit suspicious. So it never hurts to go back to IGV and take a look at the pileup of your reads and see what's going on. And what's going on here is that we have a huge pileup here. It's impossible to read this number probably, but there's 12,000 reads aligning to this spot. And if you look at the sequence, what you can see is this is a stretch of poly T. So if you remember, I mentioned that part of this experiment is to use a poly T capture piece of DNA to hybridize to the polyadenylated tail of mRNAs. So already we're thinking, interesting, this could be some kind of technical artifact. And then as you look closer, you can see that for each read that aligns to this region, really the only thing that's aligning properly is the T's. Even on reads that have more than just T's, what we see happening is this extra stuff doesn't align to the genome at all. It's all being soft clipped. So to me, this is a smoking gun of a problem. Another example, this gene ROSJEF1B, another region that is poly-T in the genome. And you see again, 10,000 reads aligning to that spot. Another example, link 00486. See this one pop up because it's a huge region of poly-G. All these reads that you see just have tons of Gs and they just get aligned here. The solution to this issue, which I fully believe to be a technical artifact, is to trim these reads before mapping to the genome. So in practice, what I like to do is to use the tool called cutAdapt to trim homopolymer sequences from the ends. So for example, this read, which ends in a long stretch of poly A, would become this. In addition, trim off these library adapter sequences. There's a template switching oligo as part of the library prep, which I didn't have time to cover in detail, but this read, when it comes up, is also an artifact of the way the library is prepared. Do others recommend this? The Hemberg Lab course that I mentioned at the beginning does mention read trimming, but many people in practice who I've talked to, many people, papers that you see in the literature, they don't mention read trimming. A lot of people don't do it, but I would very strongly recommend it. And in fact, the newest version of the Cell Ranger software, which is a software put out by 10x Genomics, it's very convenient for creating count matrix data. The newest version, only a few months old, did add certain read trimming and saw great benefits of this procedure. So it seems like 10x Genomics has caught on to this as well. And it really does seem like a valid procedure. I would recommend it. And the read trimming I've recommended goes a little bit above and beyond what Cell Ranger does in 4.0. And I think the extra bit that I do is justified. But take a look at your data, look in IGV, see if things are piling up on a stretch of homopolymers in a way that looks like it's just implausible and probably a technical artifact.
Stephen, before you transition to the next sentence, I just want to follow up with another question that I think comes back to this point about loss of information in terms of sampling only a subset of the molecules in a given cell. So the attendee asks, what biases exist, for example, most widely expressed mRNA or cell types, et cetera, that are compensated or potentially not fully compensated by efforts to process the data?
Let's see, I don't know if I can give a great answer, but for example, there are a lot of things that depend on the exact details of your experimental procedure. So if you're if you're isolating cells from a tissue, for example, like in the precision cardiology lab, sometimes we work with data from the aorta. Different tissues have different levels of difficulty in terms of liberating cells from the extracellular matrix material. Some tissues dissociate easily. Some are single cell by nature, like blood. Not hard to separate the cells. But from the aorta, it's very difficult to get cells out of that tough extracellular matrix. So you have to think about the tissue prep. Sometimes you're going to miss some cell types because they're just difficult to isolate. In terms of, and you have to think about things like cell size, for example. So in the precision cardiology lab, when we look at heart data, cardiomyocytes are a cell that's so large, it can't fit in these types of droplets. So if we just went along with the typical protocol, we would probably miss all of these cardiomyocytes, which are too big to fit in the droplets. So you have to think about these kind of things. What we do to get around that is we actually do single nucleus RNA sequencing, where the nuclei can all fit into these droplets regardless of cell type. So we can get a better representation of all the cell types in our data set. But it's something very important to think about. And for each different experiment, you should think carefully about it.
Thank you. And there are multiple questions about trimming. So I will try to summarize them into one question so that you can continue the next part of your talk. But so what is your sense of why most people or many people opt not to trim? Is there a minimum length of sequence after trimming that you keep? And do you have any preferred methods for additional trimming, either as one might use for bulk RNA, or are there any that are special for single cell work?
Great question. So I don't know if there are any tools that are specific for single cell work. I use a tool called CutAdapt, which I believe was developed for bulk work. I try to trim stretches of homopolymer from the ends of reads, and I'll have to recheck and see exactly what I do. I believe if the read ends up being shorter than about 20 bases, I throw it out entirely because it becomes difficult to map in that case. But this is a bit more trimming than what the newest version of CellRanger is currently doing. CellRanger is only trimming poly A from the 3' end and this adapter sequence from the 5' end. I assume that they're doing that just to be as conservative as possible and say, well, we really don't want to trim out anything that we don't have a rock solid justification for trimming out, which is a rationale I can completely understand. But from my observations of many data sets, it does seem there are other types of technical artifacts that lead to these homopolymer stretches in reads. And if you do sort of an in silico experiment where you trim and you don't trim, compare the count matrices that you get, see what the differences are. And I think you'll be satisfied that read trimming is a useful idea, and that the things that change in the count matrix are things that if you look at IGV, they probably should be changing. But I would encourage you to try it both ways and see what the difference is.
Thank you. And there are many questions. I think maybe in the interest of your being able to go on to the next part of your talk, we'll save those till later, but please do continue posting them. That's to the audience, and we'll do our best to answer them. So thank you.
Okay, great. Yeah, glad to have questions for sure. I will, if we don't get through everything, that's okay. I tried to pack this full, so I may have been a little overly ambitious. We'll see how far we get. The transcriptome is worth a quick mention, especially if you're working with non-human data and non-mouse data. The human transcriptome is in pretty good shape. I've heard anecdotally that the mouse transcriptome is also in very good shape. I haven't worked with mouse data myself. But if you're working with other types of non-human data, for example, I've done a lot of work with the rat, it's worth checking the transcriptome. Check the pileups of your reads in IGV, see where they align. For example, are there peaks, are there pileup peaks of reads that lie just beyond the 3' end of a gene annotation? So for example, we have a gene that's defined here. We have a pileup of reads, some of which overlap the gene and some of which don't. This particular technology that I'm talking about is 3' capture. You're capturing the polyadenylated tail. So most of the reads you get are toward the 3' end. It's common to see this kind of thing where a big pile of reads will be missed because the gene annotation is just a little bit too short. So maybe you need to think about augmenting your transcriptome if that's happening to you. Also, are there lots of suspicious annotations? If you're working with something where the transcriptome or the reference genome is in bad shape, and there are lots of contigs, you want to make sure there isn't redundancy, because if reads can multi-map to different places in the genome, a lot of times those reads get thrown out, and you can lose a lot of data that way. But this is mostly an aside about non-human work. Also anecdotally about the transcriptome, a lot of the gains in Cell Ranger software version 4 come from updates to the human transcriptome that are along these lines to try to get more reads, and they do indeed get more reads aligning. So quantification itself, software tools that are good for this. I personally have used Cell Ranger count quite a bit. I'm a fan of that. Alevin is another nice principled tool. There are several more. And just as an aside, being part of the data sciences platform, I like to do a lot of analyses in Terra, which is a cloud-based computing environment where you can define these workflows like a Cell Ranger count workflow. You can have your 50 data sets and just run them all in parallel on the cloud. It becomes quite easy to do bulk analysis, like batch analysis that way. So let's take a look at the count matrix once we have quantified it. We have our count matrix here. What if we just sum over all genes and get one number per droplet, the UMI counts per droplet? We can plot that over here. It's typical to do that on a log-log scale, which is kind of unusual if you're not used to looking at a log-log scale. But what this plot is showing, this is just plotted as a monotonically decreasing function of UMI counts. So these barcodes are sorted in order. So droplets on this x-axis. And what you can see is that a lot of the droplets have a lot of counts. So 10,000, 20,000 counts per droplet. These we assume to be good cells that were captured in the experiment. And then down here you see a large plateau of maybe 100,000 barcodes that had maybe 100 reads. These droplets were empty. And I'll touch on that just a little bit more, but this is kind of what you expect to see. A lot of cells, a drop off in this plot to a plateau kind of level. So it's worth mentioning sample level quality control. I'm going to have to breeze through a couple of these things, but it's worth looking at the UMI curves for different samples. This is an example of something that looks good. This is an example of something that looks pretty bad. Instead of 20,000 counts per cell, we have less than 1,000 counts per cell. Here we can tell what's a cell, what's an empty droplet, more or less. Here we really have no idea. It's just a smooth decline into oblivion. That's not what you want to see. If you see something like this, you really want to think hard. Do we want to use this data, or did something just fundamentally go wrong during the experiment? Also useful to plot. For example, if you use Cell Ranger count, plot all the QC metrics that come out of it for all the samples, where each dot here is a sample. And just take a look. Are some samples consistently outliers on these metrics or not? Another useful thing, actually a t-SNE plot can be produced by Cell Ranger count automatically. You can just take a look at that plot that they produce and see, is there some structure? Does it look like there are cell types and clusters? Or does it look kind of suspicious where there's just a big blob and not much distinction, not really much information about cell types in that data set? So unfortunately, sometimes you have to throw out a sample. Okay. Now on to this step, cleaning up the count matrix. After you have that count matrix, say from Cell Ranger count, what do you do before going into making a map? Let's talk about removing background noise from the count matrix. What background noise? What am I talking about? So we saw in the UMI curve that there were cells and there was this plateau of empty droplets that had maybe like 100 counts per droplet. In some experiments more, some less. So what's going on? Why are those there? That's the phenomenon of ambient RNA. Ambient RNA is cell-free RNA that gets packaged up into droplets, either on its own or along with a cell. Every time you have a cell packaged into a droplet, you also get a little bit of mRNA that's sampled just from the ambient soup of cell-free RNA that's present in the experiment. And you have some of these empty droplets as well, and you see those in the experiment. So How does this manifest in the data? How do we know this is happening? What do you see? In heart data, for example, you see these two specific markers, titin ryanodine receptor, that are specific markers for cardiomyocytes, and we really know that from biology and the previous literature. You see these markers show up in all cells, maybe just a few counts, but they are in all cell types, which is not something we expect to see. Another thing that drives this home is if you do an experiment that's a mixture of human cells and mouse cells, you expect to capture one cell per droplet. So you either have all human transcripts or all mouse transcripts. But in reality, what you see is a mixture where you have some mouse counts in human cells and vice versa. That's this phenomenon of ambient RNA. That is what makes up some of this background noise. So I need to speed up a little bit. This is a project that I've worked on with Murtash and Murtash's group and the data sciences platform called cellbender remove background. This is a tool that we have come up with to address this problem of removing ambient RNA from the data set, together with our co-author John Marioni on this project. It's a principal generative model of single-cell RNA-seq count data plus noise. And in practice, it's a command line tool that you can use to remove this kind of background RNA. What we like about it is that it's unsupervised, meaning that you don't have to tell it a lot about the data. You don't have to do a lot of analysis beforehand and figure out what's the background, what's contaminating what. You just give it the raw count matrix in, it gives you a clean count matrix out. Check it out if you have time. It's available on Terra. We have a little preprint up on bioRxiv. An example of the kind of thing that it can do by removing this ambient RNA is as follows. This is a publicly available data set from 10x Genomics, PBMCs. What you can see on the left hand is the raw data. Lots of different cell types and marker genes for each cell type, shown here as a dot plot. Prevalence of each of these genes in each of these cell types. After running this data through this tool, we can see certain trends. The one that I'll highlight here is, for example, if you look at these two genes, lysozyme being one, it seems like they're kind of expressed everywhere in all cell types here. They're popping up all over the place. And after you clean this up, because a lot of this is due to ambient RNA, what you see is that the expression of lysozyme becomes much more specific to just a few cell types. So that's just one brief little example. The tool also helps you determine which droplets contain cells. It's not always clear cut to tell where are the cells, where are the empties. Here in the green, you can tell these are all cells. In the red, those are empty. But what's happening in between? How do we know what's a cell and what's not? This tool also, as part of its probabilistic model, gives a posterior probability that a droplet is not empty. So for example, an output here where these are all droplets that contain cells, these are droplets that have 0 probability of containing a cell, and in between, it'll make a call for each droplet about how likely is it that it's a cell or not. Okay, but not all of these non-empty droplets that are discovered here are great quality. We need to apply further cell quality control. What does this cell quality control look like? This is the most artisanal part of single-cell RNA sequencing. And I usually use the word artisanal in a good way. In this case, it's not a great thing to have part of data analysis be an artisanal procedure. This is probably an area where the field needs the most work, to be honest. Why do we need to quality control cells at all? Well, if we don't throw out anything, you get a lot of empty droplets, you get a lot of dead or dying cells, depending on the experiment. You can get a lot of doublets, where the ideal scenario is to capture 1 cell per droplet, but in reality, sometimes you capture 2 cells per droplet. So you get doublets that look kind of like a smear of the expression of one cell type in another. So what does this cell quality control procedure do? It goes from, at the most basic cell QC, maybe we have a UMAP that looks like this, where it's kind of confusing. What's a cluster, what's not? What is this X shape in the middle here? What is this blob? Turns out if you take a careful look, it looks like this is dying cells. It looks like this is maybe combinations of two cell types. If we do a little bit more careful cell quality control, the very same data set can end up looking like this, where we get a much more clear interpretation of what's going on. What are the different cell types? So what are we trying to eliminate again? It's a little bit heuristic still at this point, but the thinking is eliminating dead or dying cells, cellular debris, doublets from the experiment. And what's the strategy? The strategy that I typically use is outlier detection in QC metric space. The idea is to calculate a bunch of QC metrics, including mitochondrial read fraction, exon read fraction, number of UMIs, number of genes, a doublet score from a tool like Scrublet, and any other metrics you can come up with that you can calculate for every droplet. So for every droplet, you have a bunch of these QC metrics. The idea then is to put together all of these metrics into this QC metric space and perform outlier detection in this space. So this space is a kind of a multi-dimensional space. Here I'm trying to visualize it by plotting one metric against another. Genes against UMIs, entropy against exon fraction against mitochondrial fraction. And what you can see is that there are little blobs that kind of appear in this QC metric space. Some of them appear to be low quality cells. For example, ones that have high exon fraction in a nuclear data set, which this happens to be, and a high mitochondrial read fraction. These are low quality cells. And so using some kind of outlier detection, I use a pre-made function from the scikit-learn package in Python. You can highlight a certain piece of this metric space, of this QC metric space, and pick those cells that pass QC, and the rest, these out here, fail. That's just one idea. That's what I do. This is not a standardized thing yet in the field. What do other people do? Many people use hard cutoffs on QC metrics. For example, mitochondrial read fraction. You say, I don't want to see a cell with more than 20% of reads coming from mitochondria. People put hard cutoffs on various different metrics. That's one approach, very popular approach. Mark from the Precision Cardiology Lab, who's very experienced in this kind of thing, does the kind of outlier detection procedure I mentioned, but per cell type. It's a little bit more difficult, but probably a little bit better because these QC metrics can vary from cell type to cell type. Just food for thought, it's not an easy answer. But this kind of procedure is consistent with the spirit of the recommendations from this 2019 best practices paper, where they mentioned performing QC by finding outliers in number of genes, count depth, fraction of mitochondrial reads, considering these covariates jointly instead of separately. Okay, but I will try to pack in a few more things before we have to end. I definitely put in too many things, but when you have many data sets, it's important to consider batch effect correction. There are two schools of thought. Generally, one is to use batch effect correction to group cells, but don't touch the raw data. Model the batch later during statistical testing. Another school of thought is reach in and change the values of the count matrix so that you can align everything. I'm personally a big fan of school of thought one. Use batch effect correction to group the cells, but don't change the raw data. Model batch later during statistical testing. Some of these batch effect corrections, this one is called SCVI, single cell variational inference. What you can see is that if you just use PCA and you don't do any batch effect correction, what's happening here is that the cells here colored by batch are separating by batch. They're not separating by cell type. The cell types are actually very confused if we color by cell type. Whereas after batch effect correction, which again is just used to reshape this UMAP space so that things are grouped together, we can see that the batch variable is sort of mixed. You can see mixing between the batches. And when we color by cell type, we see that predominantly this data is separating out by cell type instead of batch. One thing that I do want to mention before we end is about differential expression as well. While it's tempting to use some of the built-in functions for differential expression that are common in these tools like ScanPy and Seurat, like a Wilcoxon test or a t-test, for example, those are often a little bit too basic for a complicated study. If you're just dealing with one sample, it's okay. But if you're dealing with a complicated study design where you have lots of samples, maybe from different individuals with different conditions, health and disease, you really need to consider something a little bit more advanced in terms of differential expression analysis. My recommendation here is using a technique that I'll refer to as lemma and voom with summation. I would recommend reading this paper by Lund and Marioni, Overcoming Confounding Plate Effects in Differential Expression Analyses of Single-Cell RNA-seq Data. A very interesting paper about how to minimize the effect of batch effects, minimize false discoveries in the context of single-cell RNA sequencing data by sort of creating a pseudo-bulk vector per sample, per cell type. Unfortunately, that's about all the detail I can go into here, but I would recommend reading this paper, taking a close look, and to use a linear effects model to count for fixed effects like batch, or tissue, or individual, or disease state, for example. This kind of differential expression, there's been a great paper out in 2018, Sunnison and Robinson, about differential expression methods that compares many different methods. Lemma and voom, this procedure comes out looking very good in this comparison. And it's a very general kind of differential expression test that I like a lot. There are many further types of downstream analysis. We haven't even scratched the surface. You can look at gene sets that are different between different conditions, not just individual genes. You can look at cell-cell communication. You can look at differentiation trajectories of one cell type with time points transitioning into a different state. There are so many interesting things to look at. I would recommend for the downstream stuff, taking a look at this 2019 paper about best practices. And just a few practical considerations. I know I'm coming up to the end of my time. It's tempting to overinterpret a UMAP. Try to refrain from doing that. Cluster definitions depend on cell QC. Just a little short vignette here about subclustering. These are, for example, all the fibroblasts in one data set I was looking at. I cranked up the clustering resolution and saw all these different clusters. Difficult to interpret biologically. It was unclear what was going on. When we revisited with the help of someone who really understands the biology, and we did very strict cell quality control, eliminating doublets, being very careful to eliminate those doublets, the UMAP totally changed. It ended up looking like this, where here's the main fibroblasts, and these little clusters on the sides are different specializations of fibroblasts that do different things. Look for replication, do validation experiments to the extent possible. So just to summarize, trim reads if you can, no substitute for good data. Try to remove ambient RNA. Think about QCing cells carefully. For differential expression, I recommend lemma and voom with this summation procedure from Lund and Marioni. And just try to do everything several ways and compare the results, checking and double checking from every possible angle. With that, I'll leave it if there's any time for questions.
Thank you so much, Stephen. That was really, really wonderful. I feel like I've learned a tremendous amount in a short time, so thank you. There were many more questions than there's time for now. And I think maybe I'll just ask you one of them, and I'm sorry to the many other people whose questions haven't yet been answered, but we'll try to find a way for those to be addressed. So the question has to do with mitofraction, which is often used for dying cells.
Yes.
And the attendee asks, however, how do we consider these in the context of degenerative disease? If the disease sample have a higher mitofraction compared to controls, how do we set cutoffs when that might in fact be an interesting biological parameter?
This is a great point. So in the literature, the idea has crept in that mitochondrial read fraction is a great proxy for dying cells. I don't know if that's been proven. And it's very important to think about for your data set, is it applicable or not? It does seem that dying cells have a lot of mitochondrial reads. That's a fact. But as you say, in your case, maybe a lot of mitochondrial reads goes along with a certain phenotype that you're interested in. For that reason, I think it's interesting to look at many QC metrics, look at the distribution over the space of those metrics, and see if there's groups of cells. If droplets cluster in little groups where you can see there's clearly a definition between cells that have a lot of mitochondrial reads, cells that have less, and the bulk of the good cells. And you can look at the distribution in that QC metric space to see where the cutoff should be.